fun activity, and then we'll be done for the day. All right, so integumentary system, it says, describe the structure and functions of the skin and the exocrine glands. Fine. All right, so the structure of the skin. First of all, what's the outer layer of the skin called? Epidermis, and then underneath it you have the dermis. What's below that? Is that considered part of the skin?
No.
And what was the other name for the subcutaneous layer?
Hypodermis.
Hypodermis. All right. Some of the functions of the skin. What were some of the functions?
Protection.
Protection's big. What else?
Blood reservoir.
Temperature control. Blood. Yeah, it's a blood reservoir. Like the dermis has blood in it. Vitamin D synthesis. Remember that. Excretion of wastes. All those things. And then it's exocrine glands. So what were the two types of glands you had in your skin? Well, for the two big ones are sweat and oil, right? And then among the sweat, you had apocrine and eccrine, right? Eccrine sweat glands. Where did you find eccrine sweat glands?
Everywhere.
Yeah, they're all over the body. They have, in terms of their shape, I don't know if I emphasize this, but it does come up in the final. They have a simple coiled tubular shape to them. That just means they're like these long tubes that twist and turn. I mean, I think we saw them on the models, but I may not have said, oh, you know, that's simple coiled tubular. That's what the shape is. Where do you find apocrine?
Thick skin.
Yeah, just like in the armpits, the groin area, the beard area in men.
And they stink.
Yeah, and they stink.
Do we need to know what cell made that? We don't need to.
What cell make what?
Like what kind of cell? Like you said, acrine is simple coiled tubular.
Oh, yeah. The other type, I mean, the apocrine's similar to that also. Yeah.
Okay.
Differentiate between thick and thin skin. Fine. So where do you find thick skin? Yeah. Palms of your hands, soles of your feet. Where do you find thin?
Everywhere
Like
else.
everywhere else. Good. What else can you say about thick skin? Thick skin doesn't have what? Doesn't have hair follicles, which also by extension means it doesn't have what?
Oil glands?
Oil.
It doesn't have the oil glands, right? Because oil glands always have to empty into hair follicles. Good. What about sweat glands? What can you say sweat glands in thick skin? More or less than thin? More, right? Think of like being nervous with like sweaty palms, right? And then also thick skin has epidermal ridges, okay, that are organized that give you like your fingerprints and your toe prints, okay? All that stuff. Which is more sensitive? Who has more sensory receptors?
Thick.
Thick also. Think about, having to use, have fine touch with your fingertips and things like that. Know the order of the epidermal strata, cells in each strata. So if you're in the epidermis, let's go from deep to superficial. What's the deepest layer of the epidermis?
Stratum basale.
Stratum basale. Anyone remember what were some of the cells that were down in that layer?
Merkel and melanocytes.
Yeah, Merkel and melanocytes. Also, too, it's an important layer because what? It's the only layer where what's going on?
Mitosis.
The mitosis. So it's replacing the other layers that are on top of it. All right, what's the next more superficial layer?
Spinosum.
Stratum spinosum. Very good. Then on top of that, what do you have? Let's assume you're in thick skin. What do you have on that?
Lucidum.
Then you would have the sorry, wait a second.
No,
Granulosum. You got me all messed up. You got the granulosum, right? And that's usually like dark purple staining or whatever. Then, assuming you're in thick skin, you have the lucidum, and then on top of that, you have the corneum, right? What's the most common cell that you have in the epidermis?
Stratified. I don't know.
What's the protein that's on the? Hold on. What's the protein that's on the outside of the stratum corneum? Keratin. So what's the most common cell? Keratinocytes, right? They're the guys that are making that stuff. What's the name of the cell that plays a role in immunity?
Langerhans
The
cell.
Langerhans cell. Good. What's the purpose? You mentioned them before. What's the purpose of the melanocytes?
Protect against the UV.
Yeah, so they're making this thing called melanin, which protects against UV light damage that could damage the DNA. All right, good. Describe and know the orders of the phases of deep wound healing. Okay. What was the first phase? So first of all, what does it mean if it's a deep wound? It's gotten into the dermis, right? It's gone deep enough that it's in the dermis, so it's going to be a more complicated repair. What was the first step in deep wound healing?
Inflammation.
Yeah, and that's basically doing what? Cleaning it up. So you're going to have increased blood flow to the area. That's called vasodilation. So you want to send more blood flow. And then you also want the walls of the blood vessels in that area to kind of separate a little bit so that's called increased vascular permeability. That's so that the white blood cells that are in the blood can kind of squeeze their way out into the tissue and basically, destroy the bacteria, the other junk that's there. That's a cleanup phase. That's the inflammation. That's what gives you the redness, the heat, the pain, the swelling, all that stuff, right? You can't really do any of the real healing part of it until you've done that phase. What comes after inflammatory? Migratory, and that is kind of just named for the fact that in the epidermis, cells in the stratum basale are kind of going into hypermitosis, and they're, making more of the cells, and they're crawling their way, or they're migrating across the wound to close it. How are you repairing the dermis? How does the dermis get repaired?
With collagen fibers?
With collagen fibers. So the fibroblasts are becoming more active. They're filling in the dermis with collagen fibers, right? What's the third phase? Proliferative, which is basically just kind of like more of the same stuff. It just intensifies a little bit more. And then finally, the last phase is
Mature.
maturation. And that's kind of when your scar goes from looking. The blood vessels become more organized over time. The collagen fibers become organized. The scar starts to look better, after weeks to months, less red, that kind of stuff. You going for a better spot over there?
No, I need to charge something.
That's okay.
Yeah, I just want to see your handsome face too.
Thanks. All right. That's the kind of sucking up that will get you an A. All right. Finally, thank you. You're a model citizen. I love it. Where are we going here? Okay. Now we're on to bone. All right. Bone tissue. Describe the histology of bone, cell types, differentiation, blah 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 blah. So, okay. So, first of all, there were kind of 3 cell types. Well, all right. Sorry. Let me back up on this. Of the 4 tissue types that are in the body, what's bone classified as?
Connected.
So anytime you have a connective tissue, you can talk about the cells or you can talk about the space outside of the cells, right? So let's talk about the cells that you got. And you remember there were basically sort of 4 types of cells that we discussed. 3 of them were kind of related. One went, became another type, became another type. Those 3 were related. But then there was one that was sort of separate from the other guys. So let's deal with the 3 that are related. First of all, you had these osteogenic cells, which are sort of like your early bone-forming cells. You see them sort of, like in a fetus or an embryo, right? But then the osteogenic cells went on, and they became what? Osteoblasts. And what do osteoblasts do?
They build up.
They build bone. And now I'll tell you, the way it's worded on the final, and this just means the same thing as build bone. It says secrete bone matrix, okay? Means the same thing. It's just a really snobby way of saying building bone. Okay. And then remember, when the osteoblasts surround themselves entirely with bone, they sort of go into semi-retirement and become what cells? Osteocytes, right? So osteogenic cells become osteoblasts, become osteocytes, which are your mature bone cells. What were the ones that were sort of separate from those 3 guys? Osteoclasts, and what do?
They break
down bone. On the final, the way it's worded is resorb bone matrix, which just means the same thing as break it down. It's very snobby on this final, the way they word things. 2 other cell types to remind you guys about. So osteocytes are mature bone cells. What's the name for mature cartilage cells? Chondrocytes. Okay, good. And then way early on, like even before you have your osteogenic cells, like when you're an embryo, you have a really early connective tissue cell called a mesenchymal cell. Those are the first cells to sort of cluster together and differentiate when you're talking about like that intramembranous ossification and that kind of bone forming going on. So they're like super early connective tissue cells, and then you go into those more like these other bone cells like the osteogenic cells, et cetera, et cetera. Describe the hormones involved in calcium homeostasis: CT, PTH, calcitriol, estrogens, IGFs. First of all, does everyone know what CT stands for in this context?
Yeah, how do you say it?
Calcitonin.
Yes, that's right.
Calcitonin.
I just want to be clear because it doesn't, there's a lot of possible things CT could stand for. Could stand for connective tissue,
Yeah.
could stand for computed tomography, right? Which is dumb. I mean, it wouldn't make any sense in this context, but just so you're aware. But it stands for calcitonin. All right. And what does calcitonin do in your body?
Tone it down.
Tones down blood calcium
calcium.
levels. All right, so it reduces or it lowers blood calcium levels. Now, I will tell you on the final, it doesn't say blood calcium levels. It says serum, S-E-R-U-M, calcium. It means the same thing. All right, don't be thrown by that. What does PTH stand for?
Parathyroid.
Parathyroid hormone, and what does that do?
It raises.
Yeah, it increases blood calcium levels. And again, it says serum. It doesn't say blood. Calcitriol. What is calcitriol?
It helps
Somebody
us.
said it. What'd you? Yeah, it's the active form of vitamin D. All right. It's not the form that you get in your skin when the sun first hits your skin. That's a really early form. It has to actually go from your skin to your liver, and then it's not till it gets to the kidneys that it's made into calcitriol. So calcitriol is the active form of vitamin D. And what does it do that helps with calcium levels in your body? Anyone know what calcitriol does?
Absorb calcium from the food.
Correct. It helps you to absorb calcium from the foods that you eat. So it'll go to your small intestine, and it'll help you to absorb the calcium from the foods that you eat. You can have a diet that's great in calcium, but if you don't have a vitamin D in your enough vitamin D in your system, you'll kind of poop it all out, right? Estrogen. Now, estrogen, if you are like me, you probably immediately think like female hormone, blah, 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 blah. Estrogen also plays an important role, and I don't know if I really emphasize this at all, but it does play an important role in controlling the timing of when the growth plates close, okay? When the epiphyseal plate becomes, what's it called when it starts to fill in with bone? It's no longer the epiphyseal plate, it's the epiphyseal line. Estrogen, in both men and women, plays a role in kind of that timing of that. So like in your late teenage years, early 20s, when you stop getting taller, it's estrogen that's playing a role in controlling that.
One of the questions I had when I was studying was when for tall people, it's not necessarily that they have lower levels of estrogen. It's more so like for both men and women. It's more so like a biological like a genetic standpoint that they're just taller.
Yeah, or it might be like in some cases it might be hypersecretion of human growth hormone, which is the one that's actually producing, making you get taller. So you mean like it's not necessarily that their growth plates are staying open longer. Yeah, no, that's true. That's true. Speaking of growing, IGFs. Did I tell you what IGFs stand for?
Insulin growth factor.
What's the correct answer? I'm giving you a second chance.
I don't know it.
That's okay.
Insulin-like
Insulin-like growth factors. I don't know what you said. It's all right. I'm just giving you the business. All right. Insulin-like growth factors. These guys are like the middleman that between growth hormone and your actual bones that make you grow. So this is more an AMP2 thing, but for some reason it comes up on your guys' final.
You should just take it out.
I should just take it out. That's above my pay grade though. I'm not allowed to make those kind of big changes. So anyway, growth hormone comes from your pituitary, and it's going to make you get taller, right? But growth hormone doesn't go straight to your bones. Instead, growth hormone goes to your liver. It makes your liver release these IGFs, and then they go to your bone and make you get taller. So they're kind of like the middlemen in terms of making you get taller. That's what IGFs are. Okay. All right, moving right on. Joints. Describe the types of joints. Know examples of each and the movements permitted by each. So remember, there's a lot of classification going on in this chapter. First of all, there were three structural classifications, right? What's it called if joints are held together with like ligaments and only ligaments? Fibrous joints, or it's more like fibrous tissue, so that's why they're called fibrous. What if they're held together with cartilage? Cartilaginous joints. And then what if they have a joint cavity? Synovial joints. So those are your three structural classifications. You're either synovial, fibrous, or cartilaginous, right? In terms of functional, or in terms of physiologic or functional classification, there were also three choices based on how much movement you get, right? Mm-hmm. What if you don't get any movement? What's that called? Synarthrosis. A little bit of movement is amphiarthrosis, and then like freely movable is diarthrosis. All right. Let's throw a few things out here. Again, most freely movable joints in the body, like the big famous joints that you think of, like your knee or your shoulder or your hip or your elbow, they're going to be classified as synovial in structure and then diarthroses in function, okay? Some more obscure ones, sutures. Sutures are a type of fibrous joint. How would you classify sutures functionally? Synarthroses, right? They're definitely synarthroses. or if you looked at something like a synchondrosy, all right? A synchondrosy, how would you classify that?
Ampy?
Yeah, that's like an amphiarthrosis, all right? It means it's held together with hyaline cartilage versus a symphysis. What kind of cartilage was the symphysis held together with? I don't remember. What's the strongest type of cartilage in the body? Fibrocartilage, so it's held together with fibrocartilage. But we spent the most time in this chapter talking about synovial joints, and those guys are almost always classified as diarthroses, right? So it says, describe synovial fluid and the types of movement at synovial joints. So, what's the purpose of synovial fluid?
Reduces friction.
So lubricate, you're both right, yeah, to lubricate the joints, reduce friction, right? So that's all super important. It also helped, anyone remember
Bring
what else?
nutrients.
It also provides nutrients for the articular cartilage that's on the end of the bones, because remember that there's no good blood vessels really inside the cartilage, but it's got to be kept alive somehow. So it's a way of delivering oxygen and nutrients there. But primarily, it's there to reduce friction and help the bones move more smoothly. All right, some particular movements. What's it called when you move a body part away from the midline? Abduction, right? Abduction. And when you move it toward the midline? Adduction or adduction. When you decrease the angle between bones? Flexion. When you increase the angle between bones and it brings you back into the anatomical position? Extension. When you increase the angle, it brings you beyond the anatomical position? Hyperextension. Good. When you lift a body part upward? Elevation. And when you bring it downward? Depression. When you turn your forearms so that your palms are facing anteriorly? Supination. And when you do the opposite? Pronation. Good. When you spin a bone around its own longitudinal axis like this?
Rotation.
That's rotation. Versus, I'm going to hear some cracks. When you do this, what's that? Circumduction. Crack. What about when you bend your ankle like you're standing on your tiptoes? That's
plantar.
That's plantar flexion.
Plantar.
And when you stand on like your heels, that's what?
Dorsey.
That's dorsiflexion. Okay. Now the way it's worded on the final, like plantar flexion is bending of the foot in the direction of the plantar surface, and dorsiflexion is bending of the foot in the direction of the dorsal surface. I mean, I feel like the things kind of give it away, but that's how it's worded. All right, next chapter we have is muscle tissue. It says differentiate between the three types of muscle tissue, anatomy of sarcomere. Fine. So what are the three types of muscle tissue?
Cardiac, smooth,
Skeletal. All right, cardiac, smooth, and skeletal. Let's talk about kind of what they look like under the microscope and some of their functional characteristics. So how can you tell skeletal muscle under the microscope?
a lot of nuclei.
Yeah, so you're both right. It's going to have striations. It's going to be multinucleated. What about the direction of the muscle fibers?
They go in one direction.
Yeah, they all run in the same direction. They kind of line up nice and neat and parallel to each other, okay? And do you have voluntary control over?
Yes.
Yeah, okay. It's controlled by the somatic nervous system, and you have voluntary control over it. Okay. Cardiac muscle. What does cardiac muscle look like under the microscope?
They have
One
those.
nuclei.
Also has striations. Yeah.
And I don't remember.
Yeah, you're about to say it.
I don't remember. I know it's something inter, but
Yeah, intercalated discs. It's got intercalated
Intercalated
discs.
I was
It's-
thinking intervertebral, but that's not the
Yeah, no, that's not right. Thank God this isn't like a fill in the blank thing. Intercalated discs, also it tends to have like one nucleus per cell. Also, they don't lay out like nice and neat and sort of parallel. They're more sort of branching, okay? So that's a difference. Also, what about function? What's the function of cardiac muscle? Yeah, make your heart beat, right? And do you have voluntary control over cardiac muscle? No, it's involuntary, right? And then for smooth muscle, what does smooth muscle look like under the microscope?
Smooth.
You're too specific. Could you be more general? Yeah. It looks, well, it doesn't have, well, first of all, what doesn't it have?
Striations.
It doesn't have striations. That's why it's called smooth. And it's got kind of this what's called a fusiform shape, where it's kind of wide in the middle and then it tapers at the ends, right? You find smooth muscle usually in the wall of hollow organs. Do you have voluntary control over smooth muscle?
No.
No, it's involuntary. Also, why? By the way, do skeletal and cardiac have striations, but smooth muscle doesn't? Sarcomeres, right? Sarcomeres are the ways that the thick and the thin filaments are organized, and you have sarcomeres in skeletal and cardiac, and that's what creates the A bands, the I bands, all that jazz. Whereas you don't have sarcomeres in smooth muscle, and so you don't get that striped pattern. Okay. Keep in mind that all muscle types have actin and myosin. Skeletal, cardiac, smooth. They all have actin and myosin, but only skeletal and cardiac have them arranged in sarcomeres. Smooth muscle doesn't have them arranged in sarcomeres. Another thing to mention about the anatomy of the sarcomere. So, what's the names of the beginning and the end of one sarcomere? So, first of all, sarcomeres are repeating functional units that you see down the length of a myofibril, right? The myofibrils are the contractile organelles that are inside muscle. And if you look on one myofibril, you'll see sarcomere, 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 just repeating down. What are kind of the boundaries of the sarcomere?
The Z line?
The Z discs, right, a Z line, right? And then in the very middle of the sarcomere, you had
Midline?
an M line, and sort of surrounding that was a little area called the H band or the H zone. Everyone vaguely remember that? That represented the thick filament that didn't overlap with any thin filament, right? What's the name for the area where you have thin filament that's not overlapped with any thick filament? That's the I band. What's the name for the full length of the thick filament?
A.
The A band, right? So just, briefly remember those. The M line that's in the middle is kind of an attachment point for the thick filaments and the myosin, whereas the Z discs are an attachment point for the actin and the thin filament. Sequence the events of skeletal muscle contraction. All right, fine. What are the odds that this purple marker here is going to work? I like how that sounded. All right. Skeletal muscle contraction. So this is going to be another sequence section. All right. So, first of all, to make a skeletal muscle contract, remember, you always need to have what happen. Will a skeletal muscle spontaneously contract on its own? You need a signal from a somatic motor neuron, right? So a somatic motor neuron will release acetylcholine into the synaptic cleft, and that will cause, basically the acetylcholine receptors will bind to the acetylcholine, and they'll open up, and they'll let sodium rush in, okay? So they're essentially, the acetylcholine, so I'll just say, let me abbreviate it. Remember ACH, I told you guys ACH is the abbreviation for acetylcholine,
Yes.
right? All right. So ACH from motor neuron attaches to ACH receptor, which is a ligand-gated sodium channel. And causes, let's see, causing it to open. I'll leave this up on the board, by the way, causing it to open. And then, in step 2, sodium ions, which way, if you open up any kind of sodium channels, which way is the sodium going to move, in or out of a cell? It's going to move in, because there's always lots of sodium outside your cell. So sodium ions move in, potassium goes out. Sodium ions move in, and I'll say, initially, if you remember, initially the signaling is on the outside of the muscle cell, right? The electrical signaling is on the outer cell membrane of the muscle cell. But what's the name of those little things that allow the electrical signal to go deep inside of the muscle cell?
Gates, the ligand.
The T-tubules, right? The T-tubules are the little passageways that allow it. So I'll just say sodium ions move in, travel along T-tubules. Now, what did that electrical signaling caused what to happen inside of the muscle cell? What did that electrical signaling directly cause to happen?
Calcium from circle something.
That's right.
Sarcoplasmic reticulum.
That's right. So that's going to cause the sarcoplasmic reticulum to release calcium. I'm just going to abbreviate SR. Releases calcium. And then what did that do? Once the calcium was released, what did that actually do? Well, it's going to trigger the muscle contraction, but you got to be a little bit more detailed. How did it trigger the muscle contraction?
It's connect with the troponin that we don't like.
Who doesn't like?
Everyone. 'Cause
Oh,
the
okay.
third
Well, I
one-
love it, so I more than make up for your hatred. Yeah. What did that do though? What binding to the troponin did what?
And then they move.
It opened up the binding
site,
site. So I'll say calcium binds to troponin, which moves. You remember the other protein?
Tropomaios.
Moves tropomyosin, binds to troponin, moves tropomyosin, and opens binding sites. Or I'll say, I think it's better, well, that's fine. Opens binding sites or unblocks binding sites. Specifically, let me add this. It's opening the myosin binding sites and opens myosin binding sites on the thin filament. Then, provided that you also have enough, what was the other thing you needed besides release of calcium to make a muscle contract?
ATP.
That's right, ATP. Then, provided that you have enough ATP, the myosin and the actin will now be able to attach to each other and they'll start pulling and get the muscle contraction. So, I'll just say actin and myosin bind. Whoops, long day. Actin and myosin. And contraction. Yes. All right, so that's basically the events of skeletal muscle contraction, or at least as detailed as they want you to know it on the final. If you stimulate skeletal muscle fiber once, you're going to get what's called a twitch contraction. And assuming you don't, so if I stimulate it here at this point, the muscle fiber is going to contract. And assuming I don't stimulate it again, it'll naturally relax, right? Now, what if I stimulate it there, and say I try to stimulate it again, like right there? Will I be able to stimulate it again right away? No, remember there is a refractory period. You do have to wait, right? But suppose I waited to out here, and then I stimulated it, what would happen?
Go again.
Well, you'd get a stronger contraction. So instead of just naturally starting to relax again, it would look something like this, and the force of the contraction would go up, right? So you're getting a stronger contraction than the first.
So
Yes.
the refractory period is only up until
It's
past the threshold?
It's about. I mean, I don't have it precisely in here, but the refractory period probably ends right about there, right?
Okay.
So if you try to stimulate it any place before that dotted line, you won't be able to. They're just not ready with their little gated channels and all that. Anytime past that, you'll be able to stimulate it again, and you'll build, and you'll get a stronger contraction than the first. Okay. Basically, what it's doing is it's causing you it's causing the sarcoplasmic reticulum to release even more calcium, and the more calcium that's released, the more interaction you can get between the thick and the thin filaments, right? The more binding sites you unblock when there's more calcium available. Okay. All right. Remember I showed you that kind of cycle of how ATP is used during the contraction. What was the name of the step where the actual pulling takes place? What was the name for that?
Power stroke.
The power stroke. All right. Now, the power stroke is the actual pulling step. On the final, though, they don't call it the pulling step. They call it rotate. So this is so snobby. Rotation of cross bridges to generate force. This means the pulling step. All right. Stop putting on airs here. We all know. We're all friends here, so we don't have to deal with that. It's just mean. Describe the order of the reactions which produce ATP in skeletal muscle. All right. So if you're running a sprint, which I don't recommend, but if you're running a sprint, or doing some activity for maybe 10 seconds, where would your skeletal muscles get the ATP from? From the muscle itself, specifically, what's the name of the kind of creatine phosphate, right? That would give you the ATP for maybe up to 10 seconds or something. If you want to use, say, your muscles for like up to a minute, where might you get it from? Anaerobic respiration, aka glycolysis. And then if you want to use your muscles like even longer, like you're running a marathon or doing something crazy like that, where would you get it from? Aerobic respiration. Okay. What does aerobic mean? It implies you need what?
Oxygen.
Oxygen. Okay. All right. Next we have nervous tissue, and it says differentiate between the neuron and neuroglial cells. Fine. So obviously neurons are kind of famous because they can do what?
Electrical.
They can send electrical signals. Neuroglia cannot send electrical signals. But let's talk about in this next section here. It says describe neuroglial cells, the types, function, ability to be excited. So first of all, none of them can be excited. They're like my students in May. They cannot be excited. Okay. Or some of you in January. Some of them never get excited. Sorry, I'm cracking myself up here. Neuroglial cell types, so let's go through these. What's the name of the ones that form the BBB? Astrocytes. What is the BBB, besides the Better Business Bureau?
Brain barrier.
You got something dirty?
Yeah, I said big booty bitches.
Oh, nice. Nice.
No, the blood-brain barrier.
The blood-brain barrier, good. And what's the purpose of the blood-brain barrier?
To protect.
Yeah, to protect the brain from potentially harmful materials that might be in the blood. You don't want it to get out. So the astrocytes help with that. What's the name of the cells that make CSF?
How do you say it?
Appendable cells. It kind of rhymes with dependable, so appendable cells. They make the CSF. What's the name of the guys that can turn sort of into phagocytic cells and help like gobble up bacteria? Microglia. Microglia.
It will come.
It'll come, hopefully. Or you'll just repeat the class, right?
No, thank you.
I would not.
Okay, let's see where we're. Well, what's the name of the guys that make the myelin in the central nervous system?
No,
Oligodendrocytes. Who make it in the peripheral nervous system? Schwann cells. Good. All right. Describe an action potential and graded potentials, EPSPs and IPSPs. So, all right. First of all, who happens first? Graded potentials or action potentials? Graded potentials always happen first, and they're always caused by some kind of a stimulus, right? Mm-hmm. It could be a chemical, a neurotransmitter that's been released by another neuron. It could be like a physical stimulus, or it could be light waves or sound waves, or there's a variety of different stimuli. They cause graded potentials. Now, if you open up stimulus-gated sodium channels, what kind of a graded potential are you going to get? Depolarizing or hyperpolarizing?
You said sorry.
Depolarizing. Or, again, if we were specifically looking at a postsynaptic neuron, we would call that a what? Instead of calling it a depolarizing graded potential, we can just call it an EPSP, right? On the other hand, if you have a stimulus that opens up a voltage-gated potassium channel, what kind of a graded potential are you going to get? A hyperpolarizing, or again, we would call that a IPSP if we're looking at the postsynaptic one. So graded potentials always have to happen first. And then you get, if you reach what? You'll then get an action potential. If you reach threshold, what was the value for threshold?
Negative 55.
Yeah, negative 55 millivolts. By the way, before any of this even happened, what was the membrane potential?
Negative 70.
Negative 70, and that's known as the
Resting
membrane potential. Why was it so negative?
Because of
There were three reasons.
Sodium.
Uneven distribution of ions, and that mostly had to do with the fact that you had more potassium leak channels than sodium leak channels. What else?
the sodium and potassium.
The sodium-potassium pump, which is constantly working. Remember, that pump is putting 3 positive things out, but bringing in only 2 positive things, so that's creating a discrepancy. And then one more thing.
The anions are too big.
The anions that are kind of mostly protein anions that are trapped inside the cell. Those were the 3 reasons for it. All right, anyway, supposing you hit that, what if you have graded potentials and you hit the negative 70 perfectly? What are you going to get? I mean, if you hit the negative 55 perfectly, what are you going to get?
Threshold.
How many?
One.
1, right? What if you don't quite hit it?
It's a sub-threshold.
Nothing happens. And if you go above it,
One.
you get multiple, right? Now, does the size of graded potentials change based on the stimulus?
Yes.
Yes. Does the size of action potentials change based on the stimulus?
No.
No. You either have them or you don't. And what's that principle called?
The all or nothing.
All or none principle. Yeah, or all or nothing, I can't what to call it. All right, next it says sequence the events of an action potential. Fine. Here's another sequence section. So, so with this one, I'll just draw a little picture. They're assuming that you've got a synapse between 2 neurons. So here's your first neuron, right? And then here's your second neuron. Okay. And the starting point for this series of questions is they're assuming that you're at the very ends of the presynaptic neuron. So you're down here, okay, at the end of the presynaptic neuron. What happens at the end of so let's assume the electrical signal is going down the axon of the first neuron. What happens when that action potential gets to the end of that first neuron?
Calcium is released.
Thank you. So calcium is going to rush in. It's not really released, but calcium is going to rush in to the synaptic end bulbs.
To release.
Yep, you're getting there. Hold on. Calcium rushes into end bulb of, I'll just say presynaptic neuron. All right, and when the calcium rushes into the synaptic end bulb, what does that cause the presynaptic neuron to do?
Release a neurotransmitter.
It's going to release neurotransmitter into, what's the name of that space there?
The synaptic.
Synaptic cleft or synaptic gap, right? So we'll just say releases fine. All right. Now, the neurotransmitter is going to then cross the synaptic cleft, and then what is it going to do on the postsynaptic neuron?
It's going to go to a ligand.
Yeah, it's going to attach to basically ligand-gated channels, which are essentially receptors for that neurotransmitter, right? So I'll just say neurotransmitter binds to receptor. Whoops. And I'll just in parentheses put ligand gated channel. On postsynaptic neuron. Good. All right. And let's assume for this example that it's a ligand gated sodium channel. Okay? So it's opening up a ligand gated sodium channel. And again, what kind of a graded potential is that going to cause to happen if it's sodium?
Oh,
Depolarizing.
Depolarizing. And the fact that this is the postsynaptic neuron, we would call it a
EPS.
ESP. EPSP. Yep. So we'll just say channel
EPSP.
opens, sodium rushes in, you get EPSP. All right. Now, let's assume, well, first of all, here's another question. Is one graded potential usually enough to reach threshold?
No.
No. However, graded potentials can do what to try to reach threshold?
Summation.
Summation, right? They can add together to become stronger and potentially reach it. So let's just say there were a bunch of these guys going on. So I'll just say EPSPs add together, and I'll put in parentheses summation. Threshold is reached. All right. So you've reached threshold, yay. So that means you're going to get an action potential, right? Now, what's the first? So remember, with an action potentials, you know, they sort of look like this. They go up, and they reach a height, and they go back down. Okay. When you're on this side of the dotted line, when you're going up, anyone remember what the name of that phase is of the action potential? When you're going from your -70 and you're going all the way up, and
Depolarizing?
yeah, that's the depolarizing phase. Okay, so if you hit threshold, it starts with. And anyone remember what kind of gated ion channels are responsible for action potentials? They're not ligand gated ion channels. They are
Sodium.
voltage gated ion channels. So we'll say voltage gated sodium channels open first.
Because it would be electrical.
And I'll just draw an arrow and says that's going to be the depolarizing phase of action potential. And then, as you go on this, as you reach, usually, remember this value here, like right around there was like 0, and then down here was -70, and you kind of reach sort of a maximum of like +30, right? But once you reach that peak, then it starts going back down again. What's that phase called where it goes back down again? Repolarizing phase, and that's due to what kind of voltage-gated channels opening? Potassium. All right, so voltage-gated potassium channels open. And you get the repolarizing phase. So that's it. That's about as far as they wanted to take that. Again, I'll leave both of those things up there. Now, one other thing to be aware of with electrical signaling in neurons, which was similar to what we just talked about in muscle, is there is a waiting period where you can't stimulate a neuron again, no matter how hard you try. What's the name of that waiting period that where no matter how hard you try, you're not going to get another electrical signal?
Absolute.
That's the absolute refractory period.
Absolutely.
What's the one called where, yes, you can get a second signal, but you got to use a stronger stimulus than normal? That's the something refractory period. That's the relative refractory period, all right? Things aren't quite back to normal yet, but they're normal enough, like the channels have reset themselves enough that you can get another electrical signal. You just got to use a stronger stimulus than normal. All right, moving right along. So we have chapter then 13, spinal cord and spinal nerves. Describe the anatomy of the spinal cord and spinal nerves, the opening for the spinal nerves, the meninges. Fine. So what was the kind of landmark for where the spinal cord began? The foramen magnum. And where did it end?
The clunches.
L2,
Well,
L3.
the conus medullaris,
Yeah.
it's kind of at the L2, L3 vertebrae level, okay? That's where it ended. Now, even beyond where the spinal cord ends, what's that thing kind of hanging off the end of the spinal cord? Yeah, the cauda equina, or the horse's tail, right? So don't forget that.
That's just the root for the.
If you look on the, if you take a cross section of the spinal cord, you have white matter and gray matter. Where's the gray matter? It's on the inside, and the white matter's on the outside. Okay, so remember that. Now, the ways that the spinal nerves are connected to the spinal cord is by using roots. Okay, what type of neurons do you find in the dorsal root?
Sensory.
Sensory. What do you find in the ventral root?
Motor.
Motor. Good. How can you tell the dorsal from the ventral root?
Dorsal.
The bulge, right? The dorsal root ganglion that's sitting there with all the cell bodies of the sensory neurons that are coming in. As soon as the dorsal root and the ventral root come together, then you can kind of point and say, hey, there's the spinal nerve, okay? Spinal nerves are classified as mixed nerves. What does, and we sort of came up with cranial nerves, what does the word mixed mean in this context?
Sensory and motor.
It's got sensory and motor axons in it, okay? All right. Now, when the spinal nerves, well, first of all, what's the name of the hole in the middle of the vertebrae where the spinal cord sits? Well, that's the vertebral foramen where the spinal cord sits, right? But when you look on the sides where the spinal nerves come out, what are those called? Intervertebral foramen,
Yes.
right? Meninges. So what's the innermost meninge?
Pia matter.
The pia mater. What's the middle layer?
Arachnoid.
Arachnoid mater, and then the outer layer is
Dura
dura
mater.
mater. Okay. What's the name of the space that you have below the arachnoid mater?
Subarachnoid
space. True or false? That space is hollow and nothing's in there. False. What's in there?
CSF.
CSF. Good. Talking about snobby ways of saying things, and this was a new one on me. On the final exam, they don't use the word meninge. They use. I think this is nonsense. I gotta look this up. I've honestly never looked this up. I think it must be true. Listen, it's printed, so we know if it's printed, it has to be true. But they don't use meninge. They use meninx. I think that's supposed to be the singular form of meninge, technically speaking. Okay, all right, we got confirmation. She was quicker on the draw than you were there. So anyway, there you go. Whatever. So that, don't be confused. I mean, like I said, during the final exam, if there's any weird stuff like this, just come up and ask me. I don't have any problem telling you, like, oh yeah, that's the same thing as meninge. All right. Differentiate between a nucleus and a ganglion. Well, that's easy enough. So what's the definition of a nucleus?
It's in the central.
Yeah, cell bodies in the CNS. So a collection of neuron cell bodies in the central nervous system. Now, I will just tell you that they don't use the word collection. They don't use the word group. They use a more complicated word that maybe some of you don't know called aggregate.
What?
So
What's wrong with these people?
they're just snobs. They're snobs. They think highly of themselves. So aggregate means the same thing as like a group or a cluster of neuron cell bodies, okay? And if you're in the CNS, it's a nucleus.
Gonna be coming
If
up
you're
a
in the
lot.
peripheral nervous system, it's a ganglion, right?
Mhm.
All right. Describe the tracts of the spinal cord and the functions of each. That's actually going to come up in chapter 16, so I'm going to skip that for now. Describe the steps of the reflex arc, and the spinal reflex is discussed contralateral, ipsilateral, intersegmental. Fine. So what's the first step of a reflex arc?
Sensory receptor.
The sensory receptor. Good. What's the second part?
Sensory neuron.
The sensory neuron. What's the third part?
Interneuron.
Yeah, the integrating center, or the interneuron, or the control center, whatever you want. All of those names are probably fine. What's the fourth part?
Motor neuron.
Motor neuron. And then what's the fifth part?
Effector.
The effector. And if it's a somatic reflex, the effector will be what? Skeletal muscle. If it's an autonomic reflex, it'll be smooth muscle, cardiac muscle, or glands. All right, good. Some of the specific reflexes we discussed. Okay. So most of the reflexes we discussed were ipsilateral reflexes. What does that mean?
It happens on the same side.
That means that the sensory signal coming in and the motor signal going out are on the same side of the body, right? What kind of a reflex was the patellar reflex classified as?
Ipsilateral.
Ipsilateral, but I mean, that's called a stretch reflex, because you're essentially, for a brief moment, you're stretching the muscle when you hit, right? So that was a good example of an ipsilateral. The flexor withdrawal reflex, when you touch something painful with your hand or your foot and you pull away, that's also a ipsilateral reflex. What was the only contralateral reflex?
The cross.
The crossed extensor reflex. That's where you send signals to the opposite leg from where you experience the painful stimuli in order to support your body weight when you're trying to pull back with the foot that's experiencing like the pain or whatever. So that was the only one of a crossed extensor. That was the only contralateral reflex we had. Now, intersegmental. What that means is when the signal comes into the spinal cord, it gives off branches that go to many levels of the spinal cord. And the purpose of that is to make the response stronger. By getting more spinal cord levels involved, you'll then have more motor neurons that go out to make a stronger response, okay? It's mentioned, I think, in your PowerPoint, but I know I didn't really belabor the point, but intersegmental just means the sensory neuron goes in, gives off branches that go to multiple spinal cord levels, and then motor neurons come out from there. The flexor withdrawal reflex is a good example of intersegmental. The crossed extensor reflex is also a good example of intersegmental. Okay. Describe a nerve plexus. What are nerve plexuses, or what's the purpose of nerve plexuses?
Bunch of nerves like a branch.
Yeah, it's a way of sort of organizing neurons and creating nerves that you send to different areas of the body, right? And in terms of the plexuses that come off of your spinal cord, plexuses are made up of the anterior rami branch of each spinal nerve, and then it connects up with its neighbors above or below it, right? Do all of the anterior rami form plexuses?
No.
No. What was the area where you didn't have plexuses?
Thoracic.
Yeah, in the thoracic area, you don't really have plexuses. They just kind of go straight out to where they're supposed to go. But pretty much everywhere else you had them. What was the highest plexus, the most superior plexus that we covered?
Cervical.
Cervical plexus. And what was the famous nerve that comes off of there?
Phrenic.
Phrenic nerve. Why is it famous?
It works with your diaphragm.
Goes to the diaphragm. And what was the memory trick for it?
3, 4, 5.
Yeah,
keep
C3,
alive.
C4, C5, keep you alive. Okay. What's the next plexus you have? Brachial plexus. And that plexus is famous for controlling your what?
Upper.
Yeah, your upper extremity, right? So it's famous for that. Then you get a break in the thoracic area. Then you have the lumbar plexus. Okay. What was the biggest nerve coming off the lumbar plexus?
Femoral.
Femoral's the biggest off the lumbar plexus. And then coming off of the sacral plexus, what's the biggest nerve? Sciatic. And then there was this little teeny coccygeal plexus. All right, next we have brain and cranial nerves. It says, describe the functions of the basal nuclei, BBB, pituitary gland, and CSF. All right, so the basal nuclei are what? They're in the brain. Are they white matter or gray matter?
Gray.
They're gray matter, which is kind of weird because they're not in the cortex, right? The cortex is where you think of most of the gray matter being. But deep inside of the cerebrum, you have these other pockets of gray matter called the basal nuclei. Basal nuclei do a whole bunch of things. Okay, they help with subconscious contraction of muscles, like swinging your arms when you walk. They help you to start and stop body movements. They also do stuff that's not even related to body movements, like they help regulate emotional behavior. They help with attention, memory, planning. So they actually are involved in a lot of things. They're kind of a big deal. The BBB, again, stands for the blood-brain barrier. We mentioned the astrocytes. They're the glial cells that kind of surround it. What else contributes to the blood-brain barrier? What else makes it tough for substances to cross from the blood vessels in the brain into the brain tissue? What kind of, let me ask this a different way. The walls of the blood vessels themselves, the cells that make up the wall of the blood vessels, are held together very closely.
Tight junction.
So there's tight junctions that hold those cells together, and that contributes to the BBB, as well as the astrocytes that are like surrounding it, right? So they all contribute to that. All right, where was I here? Oh, the pituitary gland. Pituitary gland hangs under what part of the brain? The hypothalamus. The hypothalamus, and this comes up more in AP2, but the hypothalamus is considered a part of which two organ systems?
Endocrine.
Endocrine and
Nervous.
And nervous, right? Nervous because there are neurons that are in it that are traveling, but then also endocrine because the hypothalamus releases its own hormones that we discuss more in AP2. So there are direct axon connections between the hypothalamus and the pituitary that hangs underneath it, and then there's also these hormones that the hypothalamus is producing. And then CSF, again, CSF stands for cerebrospinal fluid. Normal CSF should look kind of what color?
Clear.
Yeah, kind of clear, maybe like slightly like straw colored, but it should be clear. It shouldn't be cloudy or anything like that. And where is CSF made? Yeah, inside of the ventricles. In what little areas? Choroid plexuses. By what type of glial cells? Ependymal cells. And how did they actually make the CSF? They're essentially just doing what? Filtering the blood, right? So let's walk through the pathway then. So let's suppose you're a choroid plexus and you're an ependymal cell and you make some CSF in the lateral ventricles. Where would the CSF go next?
Third ventricle?
Yeah, it's going to go through the interventricular foramen to get to the third ventricle, which is right in the middle. Then you go through what?
The aqueduct.
You go through the aqueductus sylvius, also known as the aqueduct of the midbrain and the cerebral aqueduct, right? Then you're where? Fourth ventricle. Now you have some options. If you want to stay inside of the central nervous system, you could continue downward in the what? Central canal of the spinal cord. Alternatively, you could go out through, what's those holes called? Yeah, the apertures, the median and lateral apertures. And then you'd be in what spot then? You'd then be in the subarachnoid space, right? You'd be outside the brain and the spinal cord, and then you'd circulate around outside of them until eventually, where do you get returned back to the blood?
Arachnoid.
The arachnoid villi. Okay? All right. Define decussation and projection tract. So what does the word decussation mean? It means crossing. And remember, inside the central nervous system, there's a lot of places where neurons cross sides. And that's one of the main reasons why when you have a stroke on one side of the brain, it's the opposite side of your body that's usually affected. Lots of opportunities for crossing over. Sometimes they're referred to as commissures. Other times they're referred to as decussations. And one, probably the main decussation that we talked about was in the medulla. There's something called the pyramidal decussation. So that's just one of the many places where they cross. Projection tracts. What are projection tracts? Well, first of all, what's a tract? It's a bundle of axons in the CNS. What's it called if you're in the PNS? That's a nerve, right? If you're in the PNS. All right, so tracts are bundles of axons in the central nervous system. What's a projection tract? It's going up or it's going down, right? If it's going down, if it's going up, it's a what? Sensory tract or an ascending tract. If it's going down, it's a
motor.
Yeah, it's a motor tract or a descending tract. Okay. Again, those are projection tracts. Those are what you find in the white matter of the brain, and then you also obviously find them, continuing down in the spinal cord. What's the name for the tracts that connect different gyri within the same hemisphere? Those are the ones that cross sides. Association tracts connect different gyri within the same hemisphere, and then commissural tracts connect the same relative gyri in different hemispheres by crossing sides. Know the number, name, and function of the 12 pairs of cranial nerves. Which cranial nerve has its own dermatome? Which cranial nerves are mixed? All right, fine. So make sure you review your cranial nerves again. First of all, just starting off very basically, which division of your nervous system are the cranial nerves a part of?
P and S.
The peripheral nervous system, right? Don't be thrown by the word cranial. Don't confuse that and think central nervous system. Remember what the word nerve means. It means a bundle of axons in the peripheral nervous system. So cranial nerves are part of your peripheral nervous system. Let me go through just a few things here. So cranial nerve 1 is what? Olfactory, and it is pure sensory. It does your sense of smell. Cranial nerve number 2 is the optic, and it's also pure sensory. It does your sense of vision, right? Cranial nerve number 3, 4, and 6 are related. Why is that?
For eyes.
They all move the eyeball, okay? Cranial nerve, what's cranial nerve number 5? Trigeminal. And it does sensation to your face. It also, what motor does cranial 5 do?
Opens your eye.
It also does mastication. It does chewing. Okay.
Chewing.
Cranial nerve number 7 is which nerve? Facial. That's the big motor nerve to your face. When you smile, you frown. That's the facial nerve. However, it also has sensory, right? What was the sensory thing of the facial? Taste on the anterior 2/3 of your tongue. So don't forget that. Cranial nerve number 8 is the what? Vestibulocochlear nerve, and that's going to do it's also pure sensory, and that's going to do what?
Hearing and balance.
Hearing and balance, good. Cranial nerve number 9 is the glossopharyngeal. It's going to help with things like swallowing. It's going to do taste on the posterior 1/3 of your tongue, among other things. What's cranial nerve number 10?
Vagus.
Cranial nerve number 10 is the vagus. What did the word vagus mean? Wanderer, because it goes to more places than in the body. It goes down like as far as your large intestines even. I mean, it really travels. It doesn't just stay in the head and neck area. It really is traveling pretty far. And it's a big player in your parasympathetic division. It does sensations from your internal organs. It monitors your blood pressure. It monitors like chemical levels in your blood of like carbon dioxide and pH. So it's going all over the body. It's a really big player there. What's cranial nerve number 11?
Accessory.
Accessory. What does it do?
Help you move your neck.
Yeah, it's basically having you move your head, And then lastly, what's the last one, 12?
Hypoglossal.
Hypoglossal, and what does it do?
Help you move your
It helps you
tongue.
to move your tongue, okay? All right, what else was I going to say about it? Which cranial nerves are the ones that are, I think, that comes up later. You know what, I'm not going to answer that. Which cranial nerves are considered mixed cranial nerves?
That are both sensory and motor.
Correct. You have to go, some
5.
say, Mary, 5, I like
7.
that. 5, 7,
9, 10.
9, and 10. 5, 7, 9, and 10. All right. All right, let me go on now to chapter 15, the autonomic nervous system. So it says differentiate between the somatic nervous system and the autonomic nervous system. So what can you say? So both are going to have sensory and motor branches to them, right? What can you say about the sensory part of the somatic? Are you consciously aware of sensory somatic?
Yes.
Yes. What can you say about the sensory part of autonomic? Are you consciously aware of it? No, it's subconscious. What can you say about the motor part of the somatic? Can you voluntarily control it?
Yes.
What can you say about the motor part of the autonomic? Can you voluntarily control it? No. Can't. It's involuntary. And again, that's because somatic motor always goes to what kind of an effector? Skeletal muscle. Autonomic motor goes to smooth muscle, cardiac muscle, or glands, right? Differentiate between sympathetic and parasympathetic divisions of the autonomic nervous system. So again, what's the nickname for the sympathetic? Fight or flight. Anyone remember where did the sympathetic division originate? Or what was the other nickname for the sympathetic division? Thoracolumbar, right? Because that's where you find the cell bodies of the preganglionic sympathetic neurons, right? Either in the thoracic or the lumbar part of the spinal cord, right? Also keep in mind that it's the somatic nervous system that has all the, it's the sympathetic nervous system that has all those weird connections for how it meets up with the preganglionic and the postganglionic. It's the sympathetic division that uses the rami communicantes to kind of leave the spinal nerve and then, you know, go and make those connections. Remember, you had two choices for ganglia. You could either use the vertebral trunk ganglia or the vertebral chain ganglia, or you could use the prevertebral ganglia. Okay, those were the options. Also, too, remember you could use no ganglia and instead go to what gland? Adrenal gland. You could go like straight to the adrenal medulla, which is the middle part of the adrenal gland. Parasympathetic, on the other hand, what's the nickname for it?
Cranial.
Rest and digest. And where does it originate?
Cranial and
Cranial.
sacral.
Craniosacral is the division. Now, the corticobulbar tract is something that's kind of up handling the cranial part of things. So it's craniosacral. That's where you find the cell bodies of the preganglionic parasympathetics, right? And be aware that for the sacral part, the parasympathetic system uses a set of nerves that are called the pelvic splanchnic nerves. For the cranial part, that's where you have parasympathetic neurons being parts of some of your cranial nerves. Which cranial nerves had parasympathetic branches in them? 3, 7, 9, and 10, right? Make sure you review some of the body changes that happen when you turn on the sympathetic and the parasympathetic. It's pretty simple, right? Whatever one does, the other's going to probably do the opposite, right? So what does sympathetic do to your heart rate? Speeds it up. What does parasympathetic do? Slows it down. What does sympathetic do to the size of your pupils? Yeah, dilate. What does parasympathetic do? Constrict. What does sympathetic do to your GI tract?
Drops it.
Yeah, turns it off basically. And what does parasympathetic do? Turns it on or stimulates it, right? Differentiate between preganglionic and postganglionic neurons, cholinergic neurons, nicotinic and muscarinic receptors, and adrenergic neurons, alpha and beta receptors. All right, so remember, there's two neurotransmitters that the autonomic nervous system releases. It's either acetylcholine or norepinephrine. If you release acetylcholine, you're called a cholinergic neuron. If you release norepinephrine, you're called an adrenergic neuron. Now, in order to respond to those neurotransmitters, you need receptors. And this is where it got a little bit tricky because there were two types of receptors for the acetylcholine, and there's two types of receptors for the norepinephrine. The two types for the acetylcholine were nicotinic and muscarinic. For the adrenergic, there were the alpha and beta. And then it even got more complicated because there were subtypes like alpha 1, alpha 2, beta 1, beta 2, et cetera, et cetera. So let's go through a little bit more detailed information about both of these. So let's talk about the preganglionic neurons first. If you were to look at the axons of preganglionic neurons in both divisions, what could you say about the axons of preganglionic neurons in both divisions? They have what? That's true. They release acetylcholine. That's absolutely true. But looking at the axons themselves, they are myelinated. Okay, preganglionic neurons in both divisions have myelin. They release acetylcholine, and it's the preganglionic neurons that are going to have their cell body someplace in the central nervous system. It'll either be in the thoracic and lumbar part of the spinal cord if it's sympathetic, or it'll be like in your brainstem and your sacral spinal cord if it's the parasympathetic, right? They always release acetylcholine. Fine. Then you have the postganglionic neurons, right? Now, if you look at obviously on the dendrites and the cell bodies of the postganglionic neuron, they're going to need to have acetylcholine receptors, right? What type of acetylcholine receptors did you always have on the dendrites or cell bodies of the postganglionic neuron? You always had the nicotinic acetylcholine receptors, okay? All right. Now, if you then follow the postganglionic neuron, if you're in the parasympathetic division, it's fairly easy. The postganglionic neurons, well, first of all, let's talk about the axons. If you looked at the axons of the postganglionic neuron, they're usually what? Unmyelinated. And if you're in the parasympathetic division, postganglionic neurons always release what neurotransmitter? Acetylcholine. So if you're in the parasympathetic, postganglionics always release acetylcholine. If you're in the sympathetic, it's a little trickier. Sympathetic postganglionic neurons usually release what neurotransmitter? Norepinephrine, unless they're going to sweat glands, and then they release acetylcholine. Okay. Now, if you then look at the effectors, anytime you have a acetylcholine receptor on an effector, what type of an acetylcholine receptor is it going to be? Muscarinic. Okay. If you look at a norepinephrine receptor on a effector, that's where you have options. It could be alpha 1, beta 1, alpha 2, beta 2. There's a lot of different possible options, right? All right. Next is chapter 16, sensory, motor, and integrative systems. It says, describe the structural classification of sensory receptors. So there were like 3 structural classifications. What is the simplest type of sensory receptor possible?
Dendrite.
Just the bare dendrites of the sensory neuron. That's the simplest, right? One step up, and for example, like sensory receptors for pain or temperature, they have just like the bare dendrites. That's a good example of that. One step up in complexity would be if you took those dendrites and you just kind of wrapped connective tissue around it. What are those type of sensory receptors called? Those are encapsulated, like nerve endings, okay? And what would be some good examples of sensory receptors that have encapsulated nerve endings? Not Schwann cells, but so it's Meissner's corpuscles or Pacinian corpuscles, right? Those are good examples of encapsulated nerve endings.
Is that Ruffini too?
Ruffini would also be an encapsulated nerve ending. And then finally, the third and final option for the structural design of a sensory receptor would be if you had an entirely separate cell, separate from the sensory neuron, being the receptor. And what was a good example of where you had that situation?
Gustatory cell.
So gustatory cells with your sense of taste are a good example. Also the Merkel cells that are like in the skin that do like that really fine touch. That's another good example. All right. I don't know why I wrote this, but I'm just going to say it. Thought is not a sensory modality.
What?
Thought is something, an integrative function that you do in your brain, but thought is not a sensory modality, okay? Sensory modalities include, touch, temperature, pain, itch, tickle, vision, hearing. I mean, those are all obviously sensory, but thought is not a sensory modality. Remember that on Wednesday. All right. Describe the somatic sensory pathways, posterior column medial lemniscus pathway. By the way, on the final, it's abbreviated PCML, okay? Anterolateral / spinal thalamic pathway, so that's the same pathway, it's just got 2 different names. And then trigemellothalamic, so those were all sensory. And then you had the motor pathways, which included the direct motor pathways and the indirect motor pathways. So let's go through the sensory guys first. So the PCML, it's called that because when you're in the spinal cord, it's made up of the posterior column of the spinal cord, and that included the gracile fasciculus and the cuneate fasciculus. Once it gets up into the brain, it's the same pathway, the name just changes, and it's called the medial lemniscus up there. Anyone remember what kind of sensations the PCML did?
Touch.
It does like fine
Vibration.
touch or conscious proprioception. Remember, those were some of the things that were carried by it. All right, anterolateral / spinal thalamic pathway. What sensation is that famous for? Pain.
Touch.
Okay, it's most famous for pain, but it also does temperature, itch, tickle, so-called crude touch. Okay. Now remember, the PCML and the spinal thalamic pathways, or tracts, or whatever you want to call them, they're handling sensations on most of your body. So your chest, your back, your arms, your legs, your abdomen. Those sensations are either handled by the PSML or the spinal thalamic tract. So there's two pathways essentially that have to cover all the sensations. If you're talking about just your face, there's one tract that covers pretty much all the sensations on your face. What's that tract?
Trigeminal.
That's the trigeminothalamic tract. Okay. By the way, sorry, just to go back because this was a question that was asked and I didn't really address it. This is a question that was asked in chapter 14. Which cranial nerve has its own dermatome? What's the answer? 5, the trigeminal. Remember, dermatomes are areas on the body that get their skin sensation from a particular spinal nerve or cranial nerve, okay? And remember, every spinal nerve except for C1 has a dermatome. And for the cranial nerves, only one cranial nerve number 5, has a dermatome, right? And that happens to be your face. So anyway, back to this. The trigeminothalamic tract, it is the one tract that covers pretty much all sensations on your face. Also not just on the outside of your face, but also like inside of your nose, your nasal cavity, also the inside of your mouth. Remember, you get sensations inside, you got touch sensations inside of your mouth. And you also have hot and cold sensations inside of your mouth to tell foods and things. All of that is connected up to the trigeminal. It's only the chemical taste of food on your tongue that's either connected up to the facial nerve on the anterior 2/3 or the glossopharyngeal on the posterior 1/3, right? All right. Now let's talk about motor. So motor, there's the direct pathways and the indirect pathways. The direct pathways do what kinds of movements? Conscious or voluntary movements, right? Whereas the indirect pathways do more involuntary or automatic movements, right?
Quick movements.
Quick reflexive movements or movements like swinging your arms when you walk, things you don't think about, right? Now, for the direct pathways, they all begin with the prefix cortico because they all start where? They all start in the cerebral cortex. Most of them are going to be coming from that precentral gyrus where you have the primary motor area, right? So they all start with the word cortico, and then depending on where they end, they'll either have the word spinal or bulbar. So there were 2 corticospinal tracts, an anterior and a lateral corticospinal tract. They're in charge of voluntary movements of your arms, your legs, your abdomen, your chest, all that stuff. Those are the corticospinal tracts. Whereas the corticobulbar tract was the guy that's pretty much connected up to cranial nerve number 7, the facial nerve, that does voluntary movements of your face. Okay? So those are all direct. You have voluntary movement of them, fine. Then you got the indirect. The indirect, the prefix for the beginning of each indirect tract is going to have some weird thing that's going to represent some place in the brainstem. Okay, remember, indirect pathways, they start lower. They're not starting in the cerebral cortex. They're starting someplace in the brainstem. So they're going to have crazy names like tectospinal or reticulospinal or vestibulospinal, something like that. And there were 5 of them. There's 5 indirect motor pathways, or 5 indirect tracts, I should say. What's the name of the tract? I just mentioned it, but what's the name of the tract that's connected up to the superior colliculi? That's the tectospinal tract. It's also connected up to the inferior colliculi. Anyone remember what kind of reflexes the superior colliculi were involved in? Yeah, they're involved in visual reflexes, kind of automatically turning your body and your head and your eyes to like any little bit of movement in your visual field, like, oh my god, what was that? Like looking there, versus what do the inferior colliculi do?
Based on
Same
sound?
thing, but for sound, right?
Loud.
You hear a sudden noise, kind of the startle reflex turning in that direction. All right, last chapter, we're finally there, and then I get to torture you with the game. Special senses, describe the anatomy and functions of the eyes, ears, nose, and tongue. Well, that's pretty broad, but let's start with the eyes. So the eyes have 3 layers to them. What's the outer layer called?
Sclera.
Yeah, well, it's called the fibrous tunic, but it's mostly made up of the sclera, which is the what part of your eye?
The white.
The white part of your eye. It's really tough. It's got lots of strong collagen fibers in it. except when you go on the very anterior part of your eye. What's that part called? The cornea, and that's the transparent part of that fibrous tunic, and it needs to be transparent, obviously, so light can go through. All right. Actually, sorry. Before I even get into the eyeball, let me just. I should back up a second. What's the name of the membrane that you have on the inside of your eyelids and also on top of the sclera? That's the conjunctiva. So there's conjunctiva inside your eyelids. There's also conjunctiva sitting on top of the sclera. There's lots of little blood vessels in it, and when you have when you get tired or you smoke weed or whatever, and you get like bloodshot eyes, that's from those little blood vessels that are in the conjunctiva that are on the sclera, right? All right, sorry. Back to the eyeball itself. So fibrous tunic's on the outside. What's the name for the middle layer? That's the vascular tunic, or it's sometimes called uvea. Most of the vascular tunic is made up of the what? The choroid, which is a heavily pigmented area that prevents light from bouncing around. Until you go to the kind of front part or anterior part of the middle layer, and that would be where you have the ciliary body, and that's where you'll have aqueous humor being secreted. That's also the area where you have the ciliary muscle that can contract and change the shape of the lens, right? And then if you go to the very anterior part of the middle layer, that's where you have your iris, which is the colored part of your eye, but it's also an important muscle that's in charge of controlling the size of your pupil, right? All right. Finally, at the innermost layer is the what? The retina. That's where the photoreceptors, the rods and cones are going to be. In addition to the rod and cone layer, remember you also had a bipolar cell layer, you had a ganglion cell layer, not to mention the synaptic layers where those cells synapsed with each other, so an outer synaptic layer, an inner synaptic layer, all that stuff. What was the name of the sort of exact center posterior part of the retina where you're trying to get images to land?
Macula?
Yeah, so it's the whole kind of area is the macula lutea, and then inside of that is the fovea. That's where you have your highest density of cones. It gives you your sharpest color vision, right? All right, let's talk about the ear now. You've got the external ear, right? What's this part of the external ear called? The auricle or the pinna. Then you go through, what's that passageway called? The external auditory canal, and until you come to the what?
Eardrum.
Yeah, the tympanic membrane or the eardrum, right? Then on the other side of the eardrum, you have the middle ear. And what's inside of the middle ear? The ear bones, or they're also known as the auditory ossicles, right? What's the name of the ear bone that's directly attached to the eardrum? Malleus. That then forms a joint with the incus, which then forms a joint with the stapes, right? All right. What's the name of the window that's being covered by the stapes?
Oval.
The oval window. On the other side of that, you then get into the internal ear, and there were sort of three areas or three parts to the internal ear. Where's the area where hearing is handled? That's the cochlea. Where's the area where static equilibrium is handled? Vestibule. And where is dynamic equilibrium handled? Yeah, the semicircular ducts or the semicircular canals, right? Speaking of the cochlea part itself, remember there were three sort of fluid-filled passageways that you had when we looked at those slides. The one that the fluid wave starts first in is called the scala vestibuli. Then the one where the fluid wave goes next is the scala media, which is another name for the cochlear duct. And then the third place where you had the fluid wave is the scala tympani, right? Talking about the nose, what's the area on the inside of your nasal cavity where you have your sensory receptors for smell? Olfactory epithelium. Remember, there are three types of cells. There's the olfactory receptor cells, which are also known as the olfactory sensory neurons. And then you have the ones that sort of help out called support cells, and then you have the ones that are able to undergo mitosis and replace them called basal cells, right? In terms of the tongue, you have these different bumps called, what's the name for the bumps? Papillae. What's the biggest bumps that are at the back of your tongue?
The valley.
Vallate papillae or the circumvallate papillae, right? What's the name for the papillae that are on the sides of your tongues that look like stripes?
Folate.
Foliate papillae. What's the smallest papillae you have?
Filiform.
Filiform papillae. And also, what was special about the filiform papillae?
No taste buds.
They don't have any taste buds. All right. And then the other type we had that we didn't mention are the fungiform papillae. Describe the cells involved in the special sensory pathways and into what area of the cerebrum do they project? Fine. So, the types of cells that you had with smell, taste, hearing, and equilibrium were actually all sort of the same types of cells. They're all referred to, they were all sensory receptor cells, but they were all essentially hair cells, okay? So you had like olfactory hair cells or gustatory hair cells, or you had the inner, you had the hair cells involved in hearing, or you had the hair cells involved in equilibrium. So there's a lot of hair cells that are involved in your special senses. About the only place we didn't have hair cells was with like vision, right? So keep that in mind. In terms of where they project into the cerebrum, remember like sound goes to like the temporal lobe, or vision goes to where? Occipital lobe, okay? It's just that kind of information. Bipolar cells are also seen a lot with the special senses. You had bipolar cells with your sense of smell, okay? Those are essentially the olfactory sensory neurons. They are bipolar cells. And then you also had bipolar cells when you looked at the retina, right, with your sense of vision. So they're another common type of cell that you see with your special senses. Know which sensory pathways synapse in the thalamus. Well, it's a lot easier just to remember the one that doesn't synapse in the thalamus, and that's what?
Olfactory.
That's your sense of smell, olfactory. Also be aware with smell, it's rapidly adapting. What does rapidly adapting mean?
You get used to it.
That it means you get used to the sensation very quickly. You stop sending electrical signals to your brain, even though the sensation may still, or the stimulus may still be in the room, you've stopped sending quick signals to your brain very quickly. All right, that's it.